Welcome, stay-at-homers. I guess that's the best way I could put it. We're going to be covering a couple chapters for the rest of the semester, and I'm going to outline what's going on. Appreciate your patience. It's not a trivial thing to convert a seated class into an online, as you can imagine, and I really appreciate your allowing me to have some faux pas along the way. I really have been trying to recreate the sort of the classroom experience as a take-home type of environment, so that you don't feel homesick and that you miss the classes. I know that you deeply do, but anyhow, what we're going to do is go over various chapters, and I'm going to outline that here. We're going to do this chapter 8, which is on chromosomes and cell division. Nothing like what was broadcast. I don't know where that came from, but anyhow, it's there. We're going to do chapter 9 on genes and inheritance, move to chapter 10 on evolution and natural selection, and then 15 on evolution and diversity. Now, if you notice, 8 and 9 kind of go hand in hand, and 10 and 15 do. So what I would like to achieve is that we have 4 weeks left. We will spend 8 and 9, 2 weeks worth, and then have a quiz, not an exam, but a quiz on 8 and 9, and then we'll move to chapters 10 and 15, and then have another quiz. So the quizzes won't count as much as a regular exam. We only really had plans for one more exam, and what I'll do is equally weight the quizzes, and there will be worksheets and other things to kind of fill in for those sorts of things. And I will have these very brief, short little take-home quizzes to get you ready for the regular quiz. At least that's the thinking right now. I'd love to get your feedback, if that makes sense. I hate to just do it as one, seeing how it's a transition into a sort of a different type of format. I'd rather break it up into smaller quizzes so you can readjust without too much problem. Quizzes won't be very in-depth. They will be done online. I haven't figured out the best way to do that yet, but anyhow, we will address that. So this recording that you're listening to now will be broken into 2 parts, chapter 8 part 1 and part 2. That way it'll be spread out. You can listen to them. There'll be some sort of handout or worksheet that goes along with this. It may not be posted at the same time, but it'll be close, and we'll go from there. We are going to be using the 4th edition of the book, but really we're going to rely more on the slides, but I hope you have the textbook. The chapter numbers are off in the 3rd edition, as we learned, and the 4th edition. So all you have to do is find the right chapter if you have the older book, and everything is almost the same, but you can use the slides as the guide as we go through that. Now, I've rearranged Blackboard, in the process, I should say, of rearranging Blackboard. You can see that I have in the lesson section how you click there, how to access Teams, and I'm going to have some projects and things and figure out the best way to do this. So we'll learn together. It's not something I normally do in the course of a day, but I will have another type of case study I'd love to throw out as a way to allow some collaboration type of work together, and see how that goes. It's primarily designed, it should help your grade, not hurt it, and it should be fun to some degree. But all the information for the various chapters will be documented in each of the chapter folders that you see. Now, I made the other chapters, they're down below, so they're still available because the final is comprehensive, and I still haven't figured out the best way to share the old exams yet, and I'm talking with management to figure out the best way to do that. Other than that, always issues, but hey, look, we're going to get through this just fine, so not to worry. Now, in the lab part, I've been trying to figure out the best way to do this. This is not the only class that I teach. I do a microbiology, and you can imagine issues that I'm having trying to equate a laboratory experience for my microbiology students. And it's clinical, and it's in a BSL-2 lab, which means that it has a level of security. So that makes it really, really tough for me, but we'll get it, and the students will get what they need out of it the best I can. And the same I have for our experience in 110. And so we're going to be covering Unit 10 on genetics, and that'll be posted later today. And it's part of the crime scene investigation PCR unit that we already have done. The results will be posted for those. I did keep the gels, so I just have to kind of pull all that together. And I will have a Unit 9 summary video to help you with that. So it kind of visits the whole thing on PCR, and hopefully ties that together so that you feel good about that and the results that you obtained. The gels look pretty good. I mean, you guys did good. We got the pipetting technique down and those sorts of things. And now we're going to be moving on and looking at some genetics, which we'll obviously learn about blood typing and various things like that. I usually do blood typing, but find out what your blood type is. I'd be really curious. I might do a poll, just a quick poll, just to see what everyone's blood type. You can ask your parents if you don't know. As I say, I can't do the blood typing now, obviously, but it's something that students usually are interested in, to find out what your blood types are. It's a good thing to know, and we'll talk about why that's important and some other aspects, and you'll see that when I get into the lab. And then we'll talk about evolution and microbes, are pretty much what we're going to be spending the rest of the 4 weeks covering in the lab. It should be interesting, and I really try to stay to the theme. I'll make it relevant and try to put these things together with what things that are important to you and you should know about. Obviously we'll mention something about the virus, but I don't know about you, but I've had about all I ever needed to know about this coronavirus. I'm just saturated to the point where it's enough is enough. But anyhow, regardless of that, you can find this information pertaining to the lab in the laboratory section like we normally have, and I have them arranged in the various units. We use the units of genetics, evolution, ecology, and microbes, and they'll be populated as I pull the material together. A lot of the material I have to redo because it's for obviously not being in person, and but I will be using the laboratory manual. Now if you don't have the manual, I will provide copies of the material as PDF and as a Word document, so you can enter information into the Word document and then submit those to me as to making sure that you've gotten the work and you understand the lab and the end of chapter questions and that sort of thing, and make it easy. Instead of retyping or doing any, I just convert them to Word format, and so that you can enter that data and then just email me the documents, and that way I know. And also, as you hear this, if you could send me an email just saying that I listened to the presentation or whatever, just so I can kind of get a handle on who's out there. Being obviously in the virtual world, it's hard for me to do an inventory of what my students are, what they're doing. So anyhow, that is where we are on that. Okay. Also, I have office hours from 9 to 10 and 1 to 2. It could be any of my classes, which is typical for office hours. I decided on using Zoom. It seemed to be more accommodating and faster and that sort of thing. At least for the interim, you can use the HTTPS link there that I have, and just type in. It's the same one that's in the announcement, and all you have to do is follow that link, and you immediately come right to the presentation. So you just follow in anytime during that hour. I will be on there monitoring, and you can ask questions, whatever. So that's there. If that for some reason is intimidating or you don't want to do it, it's not necessary, it's not required, it won't get penalized in any way. You can always send me an email, or you can call my Google phone at 919-331-6037, and to try to do that while, during the office hours if you could, and I will have my phone with me at that time. If it's an emergency or something and you need to get hold of me, feel free to call me, but just try to remember that I'm just trying to be around the house and creating these videos and things, so sometimes I'm recording, and I try to keep it so they turn out okay. Now, the objectives for this chapter will be looking at the different types of cell division. Now, we've gone over this in lab, but it won't hurt to go again. But I add different things in this. And one of the things that we're going to talk about are the chromosomes and some unique aspects of the chromosomes that have some interesting characteristics. And we'll talk about that. And the process of mitosis. And there are things that we need to know about it that can actually affect the way we age, for example. So we'll look at that. And sex differences in the chromosomes, which we already talked about. So this would be a good transition chapter. A lot of this you already know in some ways or form. If not, this will be a good time to pick that up as we go along. Just to review, the chromosome, of course, we have two identical parts of chromosome after the S phase, held together by a centromere, where the two chromatids touch. So each chromosome is made up of two chromatids, the two arms that you see here, and they are held together by that centromere. And then we call the long arm Q and the short arms P. It's just a standard nomenclature for that. We remember 23 pairs, 46 chromosomes. That's the karyotype of humans. This just shows that particular arrangement. What I wanted to talk about is something really interesting, is this concept about telomeres. I've alluded to it in class, I think, one time, but it's focusing on it now. It's very much like the little plastic tips on your shoelaces. It keeps the end of the shoelaces from getting frayed, and it's got to go through the hole, so if it's not kind of pointy and that sort of thing in that plastic, it starts to damage the ends of your shoelaces. So think of the telomeres as sort of where the red is there. That is the little plastic tips at the end of the shoelaces. Why do I mention that? Well, we're finding out that telomeres are important in determining age. You may look at some reports that are talking about, well, chronologically you're, let's say, 28 years old, but if you look at your telomeres, how much damage has been provided or has happened to your telomeres, and then you have a telomeric age. And they say that the telomeres get damaged at the ends are better predictive of how long you'll live. And there's a lot of science about adding the telomeres back. There's certain things that they're trying to do that help that health. Now, of course, there's a lot of spam or junk science out there that says if you do this or that, you can live forever and all that. But potentially, if we understand this more, the telomere protecting the ends of it could help one to live longer. The reason we know this, there's a particular disease called progeria, and this particular disease is called the Hutchinson-Gilford progeria. The children rapidly age with this particular configuration, and even though this child is perhaps 8 years old, his body's aging as a result of the damage to the telomeres. Now, if you think about this, this isn't one or two cells. This is all, the 100 trillion cells that we have in our body, and have kind of in sequence have gotten damages to the telomeres to some degree. and this rapid aging. We don't typically see folks living much longer than 18 years old and that sort of thing with this particular condition. So the dynamics of the telomeres really can predict how long one might live. Now I have a link there if you're interested. You can pull that off the slide set that I have if you want to learn more about this. There's a huge amount of information out there about this. If this is the first time you've heard of it, I'll talk a little bit about it. But the telomeres undergo this attrition. Attrition just means that you're losing something over time with each cell division. So telomeres length is associated with age relative, sort of what we call age-related diseases and mortality in the elderly. And as it shortens, the problem is that we get higher and higher risk for cancers and cardiovascular disease, things that tend to shorten life. And it's really quite interesting. And they've done studies looking at large numbers of people, if you can look at the N. Now we've talked about that in statistics. Large numbers of N's dictate really the age at which someone may live, but we have to have that large sample size to believe it. And look at those numbers. There is a fair amount, U.S. and other U.K. and other places, that show the length of the telomeres and the percentiles, and then looking at the average age of the lengths and things. So it's very interesting. The U.K. seems to the average years and the percentiles following with the length of the telomeres, and so you can see that there is a correlation with that and how that fits. So anyhow, it seems to be a realistic thing, and if you look at some of the percentages there, the length of 64 and 6.4, 6.6, and 6.4 in Denmark and the like. Looks like the males are the ones getting hammered here. It always seems to come down at really more and more explanation why we don't see more men in retirement homes. For some reason, we just seem to get hammered from every different side there is. But there is some hope in Israel, but it seems like the males and females are tracking. So whatever is going on different with that is something to look into. This would be a really interesting study to do. There's so many aspects to this. I'd like to know this is the same thing in dogs or other animals and things. It would be interesting. So telomere length can potentially be influenced by a wide range of factors, including parental age at time of offspring's birth, the level of inflammation. There's that word again, inflammation. It seems like every time it's always inflammation that seems to do the most damage. Tobacco smoking, physical activity, of course being male or female, social economic status, body mass index. The heavier you are, the more body fat seems to be really negative on the health of the chromosomes. If you take vitamins or not, dietary antioxidants, if you eat blueberries and natural antioxidants like that, how much alcohol you consume has really a negative impact. Hormone replacement therapy and race. All of these things are really interesting to look at in terms of the telomeres. So we've talked about just basic things, but if telomeres become too short, additional cell divisions cause the loss of essential DNA and cell death. Some types of cells that rebuild their telomeres with each division can become cancerous. So there's some really weird things going on here, and it's a really interesting study to look at. The chromosomes, we're going to talk a little bit about bacteria in lab at the sort of the end part of the lab, but prokaryotic and eukaryotic cells. The prokaryotic cells is like one huge rubber band. Its circular piece, of course, is DNA. and it's not encapsulated inside a nucleus or protected that way. It's all just kind of folded into the milieu of the cell. Eukaryotic cells, on the other hand, have a compartment of the nucleus, and it's pretty well stored in there and protected, and then anything that's made from it, as we talked about, is transported out as like messenger RNA into the manufacturing floor of the rough ER, and things move out there to be made, and proteins and various things. So we've talked about that to some degree. Bacteria go through, they don't have a male, female, or anything like that. It's binary fission. The DNA attaches to the membrane and sort of unzips into this replication, and then the cell divides or pinches, and each of the new daughter's offsprings will now encapsulate the new copy of the chromosome, and we call that binary fission. And the event can be as quickly as 20 minutes for every event in bacteria. So most bacteria and archaea, now archaea are the really old bacteria that live in extreme temperatures and various things like that, and that's all it's making reference to. But in bacteria, the chromosome is a big circular piece of DNA. It has no ends to it, and it's replicated simply by sort of unzipping the two strands and reproducing the big circular piece, and then it gets equally divided. Each budding binary cell has a copy of the chromosome, and it's an asexual reproduction called binary fission. So in eukaryotes, it's much more involved. If you recall, we had a gap 1, then we had the S phase, and then gap 2. In the normal day-to-day operations, gap 1 is just sort of the living part of the cell, and then the nucleus decides, well, things are getting kind of worn out, we need to do a spin-off, and they go through this phase where they replicate the DNA and make copies, getting ready now for a spin-off or a division event. And gap 2, that comes after the synthesis phase or the S phase, gap 2 sort of gets it ready for the actual mitotic phase to divide the chromosomes, and through mitosis we get equal numbers of chromosomes being delivered into two cells. So we've successfully done a spin-off of those. And so we have these checkpoints, and the checkpoints are really used for several reasons. One is making sure the age and the integrity of the cell is there, and then it's just a way for the cell to justify when we do the synthesis phase. It's a very expensive phase for the cell because it's got to replicate all of that genetic material. G2 phase is getting everything ready, sort of like moving, where you pack everything into boxes and everything like that, and then you finally go into the mitotic phase. But this is really what dictates how fast or how slow cells divide. And we make reference to the hair on your head, as the cells that produce the hair replicate and divide at a certain rate, producing the hair growth that we see. But we don't see the same for our eyebrows, and so that's adjusted by these checkpoints and other types of control mechanisms to adjust the growth of the cell. And this is what this slide is really kind of resolving, is showing the various phases, and establishment and control of those will really dictate the overall cell growth. So eukaryotic body cells are somatic cells. Somatic is just a fancy Latin term just meaning body cells. Alternate in a cycle between cell division and other cell activities. The cell division portion of the cell is called the mitotic phase. The remainder of the cell cycle, called interphase, consists of two gap phases which during cell growth and other metabolic activities occur, are separated by the DNA synthesis phase, during which the genetic material is replicated. So the cell cycle control functions through a series of these checkpoints, and remember, that's what controls how fast cells are. And of course, the understanding of the genetic material, I think we talked about with Francis Crick and James Watson, and they got the two-stranded nature of the DNA and the complement of the pairs of the A's and T's and G's and C's and all that, and they got charged with that understanding. And we have to understand, in cell division is preceded by the replication of the chromosome, and that requires DNA enzymes that manage the replication of the cell. And it's one of those things that we take the parental DNA, sort of unzip it, and then make complementary bases to each of those, so we get two identical daughters copies of the DNA. And because of that, being the G's and C's, if there is an error, you always can compare with the other base to know what was right, what was wrong. So it's a really interesting technology, if you want to look at it that way. The backbone structure is stronger because of the phosphate group, the types of bonds associated with that. And we know that the DNA is associated from 3' to 5'. We had done worksheets and we talked about that in previous sessions. The Chargaff rules are really, Chargaff worked out the A's and T's and G's and C's and how they're related together. So if we have A's and T's making up 60% of content, then we know that there's 40% of G's and C's on the rest. So it's just a kind of a relationship. The structure of DNA is because it dictates the directions along a linear molecule's replication, that occurs. Nucleotides have those three components: a nitrogen-containing molecule with a base and the phosphate group, which I just showed in the previous slide. Each of the 5 carbon atoms in the sugar molecules are given a number, and we have, as you remember I went over in class, we have the 1, 2, 3, 4, so we number from the oxygen as we go around the molecule, the ring structure, and we name that. So the 3' and 5' are the carbon atoms as we count going clockwise from the oxygen, and that's where the nomenclature comes from. And we have the 2 types of bases. We have purines, which the purine is a shorter word, and it has a more complex structure. That's how I remembered it. It's just strange how that is. So the purines have the adenosine and guanine molecules, and pyrimidines, which is the longer word, have the simpler structure, ribose structures that we see here, thymine and cytosine. And so you can see the differences there of the bases. And they're broken into purines and pyrimidines. And so the T's and C's are pyrimidines, and the A's and G's are purines. And again, just to review, the nomenclature of naming is the 1, 2 going clockwise, 3, that's the 3' position, 4, then that's the 5th carbon in the sequence, and that would be the 5'. And we've already gone over that, but it doesn't hurt to review. We'll go over that again. And the DNA is anti-parallel, and this diagram really pins that down. We see the 3' to 5' and 5' to 3' on the 2 sides as it goes across. And anti-parallel means that you can see the arrows pointing in this direction and in that. They're anti-parallels of one another, which really threw the scientists for a loop when they were identifying this. The DNA replication is really, really interesting. And I'm going to stop here and break the presentation up. We're going to talk about the DNA replication part. I'm going to show a video animation about how this goes, and that will be included in our video session next time. I'll be posting that probably later tonight or tomorrow for part 2 of this. And again, this will at least get you going. Listen to the video the first time, and let me know your thoughts if you like it. And please email me just for sort of a attendance type of thing, and it kind of gives me a clue of the audience out there, if anyone's there listening or not. It kind of helps to know that. So until next time, I hope this was informative, and please give me feedback if you like it or not, and to improve or not. Thank you.